Imagine microscopic scissors, so precise they can snip the very blueprint of life itself. These invisible blades operate within our cells, performing incredible feats of genetic surgery. They possess the power to rewrite our DNA, potentially eliminating inherited diseases forever. This revolutionary technology promises a future where genetic codes are no longer destiny. For millennia, the complex instructions within our DNA seemed unchangeable, a fixed genetic destiny. Scientists once believed altering these fundamental codes was an impossible dream, confined to science fiction. However, the discovery of molecular scissors revolutionized this understanding, opening vast new possibilities. These remarkable biological tools, known as CRISPR-Cas9, originated from an unexpected source: bacteria. Bacteria developed CRISPR as an ancient immune system, a defense mechanism against invading viruses. When a virus attacks, bacteria capture snippets of its DNA, storing them as a genetic memory. This bacterial archive allows them to recognize and neutralize future viral threats with incredible accuracy. The CRISPR-Cas9 system then acts as a highly specialized search and destroy team, targeting specific viral sequences. This groundbreaking insight provided humanity with unprecedented control over the very fabric of life. At its core, the CRISPR-Cas9 system employs two main components: a guide RNA and the Cas9 enzyme. Imagine the guide RNA as a microscopic GPS system, programmed with a precise DNA sequence to locate. This guide RNA is specifically designed to match the exact genetic target scientists wish to modify or remove. The Cas9 enzyme, the actual scissors, travels with the guide RNA, ready to perform its cutting action. Together, they meticulously scan the vast stretches of DNA within a cell, searching for their assigned destination. When the guide RNA finds its perfect match, it locks onto the specific DNA sequence with remarkable precision. But that's not the strangest part. This recognition triggers a dramatic transformation in the Cas9 enzyme. The Cas9 enzyme undergoes a conformational change, positioning its molecular blades directly over the target DNA. With surgical accuracy, the Cas9 enzyme then makes a clean, double-stranded cut in the DNA molecule. This deliberate break in the DNA helix is the crucial first step in any genetic editing process. Once the DNA is cut, the cell's natural repair mechanisms kick into action, attempting to fix the damage. Scientists can then hijack these repair pathways, inserting new genetic information or disabling problematic genes. For instance, they can introduce a new, healthy gene where a faulty one previously caused disease. Wait until you hear what comes next. The potential applications extend far beyond simply fixing mistakes. Researchers can precisely turn genes on or off, controlling their expression with unprecedented control and specificity. This level of genetic manipulation was unthinkable just decades ago, confined purely to theoretical discussions. The ability to edit specific genes opens doors to understanding complex biological processes more deeply than ever. Scientists can now study the function of individual genes by selectively altering or removing them in cells. This targeted approach has accelerated research into cancer, neurodegenerative disorders, and infectious diseases significantly. The system is incredibly versatile, adaptable to nearly any organism with a known genetic sequence. It's like having a universal word processor for life's code, allowing edits, deletions, and insertions anywhere. And then something unexpected happened. This tool proved even simpler and more efficient than initial theories predicted. Its relative ease of use and low cost made it accessible to labs worldwide, democratizing genetic research. This widespread accessibility has led to an explosion of discoveries and innovative genetic therapies in development. The precision of these molecular scissors minimizes off-target edits, making the process safer and more reliable. Understanding how bacteria naturally use these scissors provided the key to harnessing their power for human benefit. The implications of these microscopic scissors are absolutely monumental. Researchers are using CRISPR to engineer immune cells to better fight cancers. In human health, CRISPR offers hope for treating previously incurable genetic conditions like cystic fibrosis or Huntington's disease. Clinical trials are already exploring CRISPR-based therapies to correct faulty genes responsible for severe blood disorders. Beyond inherited diseases, researchers are using CRISPR to engineer immune cells to better fight cancers. Imagine a future where a simple genetic edit could prevent a child from inheriting a devastating illness. This technology also extends to agriculture, enabling scientists to develop crops resistant to pests and diseases. It allows for creating more nutritious plants, or those better adapted to changing environmental conditions globally. We could see food production enhanced significantly, addressing global food security challenges with clever genetic solutions. The ethical considerations surrounding germline editing remain a crucial discussion point among scientists and society. Society must carefully navigate the profound moral questions and potential societal impacts of such powerful genetic tools. Nevertheless, the ability to precisely manipulate life's building blocks represents a paradigm shift in biological understanding.
This discovery fundamentally alters our relationship with disease, evolution, and the very definition of life itself. The microscopic scissors have truly opened an entirely new chapter in humanity's ongoing quest for knowledge.
Their scissors have truly existence opened and proves that some of the entirely new greatest chapter in breakthroughs often hide in the humanity's simplest, most ancient biological systems. The story of molecular scissors, born from bacterial defense, profoundly illustrates nature's ingenious solutions and our capacity to learn. These tiny tools, unseen by the naked eye, hold immense power to sculpt the future of life on Earth. From battling ancient viruses to editing human destiny, their microscopic tales are truly astonishing and transformative. The elegance and efficiency of these biological mechanisms continue to inspire groundbreaking scientific endeavors globally. If you found this glimpse into the microscopic world fascinating, consider subscribing for more incredible scientific discoveries. And let us know in the comments which microscopic marvel you find most intriguing.